Welcome back, life science learners. I trust that you're feeling refreshed and that you've had a chance to unwind. In this segment, we're going to look at the process of protein synthesis. Let's try and understand the context of where we are. We've looked at RNA and its structure. We do know that DNA codes for characteristics in a cell. In this segment, we're going to spend some time looking at how DNA codes for protein synthesis and how that protein is synthesized. So essentially, the term synthesis refers to putting together something, constructing. So we're going to look at the process of protein synthesis. So guys, as I mentioned, we're going to look at protein synthesis and gene coding in this section. So what is a protein? If we go back to understanding what a protein is, proteins are organic molecules that are produced in the cell in an organelle called the ribosome. And this is again context to cell organization. Proteins are a very important part of a cell in terms of how it functions. We know that in a cell, enzymes are manufactured and these enzymes have specific functions. So when we talk about enzyme production, essentially those enzymes are made during a process called protein synthesis. We also know that proteins are the way a gene is expressed. What do I mean by that? A gene codes for a specific characteristic. How does that characteristic present itself? I.e., at the start of this lesson, we mentioned that, have you ever thought about how does a gene code for dark color hair? And hence, it is the expression of the gene where it sends out a specific message that is now going to be presented in a protein. And the presence of that protein in the cell will allow for what we call a physical appearance of that characteristic. So let's try and understand this complex process by looking at it and unpacking this very simply. So proteins are macromolecules. Again, DNA is a polymer. We refer to proteins as also macromolecules. They contain elements called carbon, hydrogen, oxygen, nitrogen, and sometimes phosphorus. We also know that the building blocks of proteins are called amino acids. And so we call them the monomers. And these are joined together. When we put them together, they're joined together by special bonds called peptide bonds. And these eventually form a chain called a polypeptide chain. So to unpack this, proteins are made up of monomers called amino acids. And these amino acids are present in the cytoplasm. We have a host of about 20 different amino acids. And these come from your diet. So when you're consuming your proteins, your meat, your fish, your chicken, your beans, these are rich in proteins. And so those proteins are broken down during digestion. And so we absorb all of those into our body and we have the building blocks of proteins. The body is then tasked with taking those amino acids and putting them together to make the amino acids chain that we require, i.e. a specific protein. So let's try and understand that process in detail. So the process of protein synthesis consists of two predominant steps. The first step is called transcription and the second step is called translation. I'm going to pause at the word transcript and I'm going to draw reference to what is a script. So if we try and understand what is a script could be a message, it could be information. And that information is essentially information that is having a specific code. So the first step in protein synthesis is called transcription, and the second step is called translation. If we were to understand the word translate means change the language or change the meaning. So the second step involves changing the script into a different language, or in this case, a different structure. So let's try and unpack this. So here I've got a very complex diagram with lots of information on it. We're going to try and unpack that by looking at this process in two different stages. To help you, let's understand the context of where we are. Guys, here's a cell, and that cell has been enlarged, and it's indicating that we now in a structure inside the cell which is represented by the nucleus. So if I were to illustrate where this process is happening, so we're in the nucleus in here, and on this side we outside the nucleus in an area called the cytoplasm. So just to give you context to where this process is happening. So the first process, as I mentioned, is called transcription, and that occurs in the nucleus. And that's followed by a process called translation, which occurs in the cytoplasm. Let's try and understand each one of these in further detail. So as I mentioned, transcription occurs in the nucleus, and it involves the production of an mRNA molecule. So as you would see in this process, a newly formed mRNA molecule is produced that leaves the nucleus. So this process of transcription involves the production of an mRNA strand. Guys, mRNA stands for the messenger RNA, and the production of an mRNA is to carry a specific message to the protein synthesizing factory, which is found in the cytoplasm. It's important that at this point we try and unpack the process of transcription. So when we look at transcription, transcription involves the production of mRNA. The question that you need to ask yourself is, how does this happen? This happens in the following way. So we've got DNA, and that DNA codes for sequences of information called genes. So I'm pointing to a specific gene here. So the DNA will unwind, and the hydrogen bonds break open, and that exposes the DNA strand, and that's what you would see here. So the DNA strands have opened up, exposing a template. This specific template has a sequence of nucleotides that now code for an enzyme or for a protein. We need to recognize that all of this is happening through an enzyme called Rna Polymerase. So the Rna Polymerase will be important in this process of putting together the mRNA. For the production of mRNA, we need the monomers, and if you refer back to the previous slides, the monomers are the nucleotides that make up RNA. So as the strands separate, we find that the enzyme Rna Polymerase brings in complementary RNA nucleotides to base pair with the DNA template. So, and we would see that, for an example, the guanine base pairs with cytosine, the adenine base pairs with thymine, and very interesting is that the uracil base pairs with adenine, and we see that happening twice. So this is the production of the mRNA, which occurs because of the Rna polymerase. I'm going to try and unpack that in the next slide. And as that occurs, the entire gene is transcribed, and that gene then leaves the nucleus through the nuclear pore. The DNA then winds back into its structural form, and that process of transcription is complete. So if we were to recap on the process of transcription, transcription is the production of mRNA. In order for that to happen, the cell and the nucleus will identify a specific gene. At that point, Rna polymerase is able to unwind and break open the strand, exposing a template. The Rna polymerase brings in the free RNA nucleotides to base pair with the DNA template, and in that process produces a single strand of mRNA, which codes for the sequence of that gene. That mRNA is then released, it leaves the nucleus, and it moves out into the cytoplasm. The DNA then recoils, and it reconfigures its structure back to a double-stranded molecule. The mRNA that leaves now has the information, and that mRNA has the information in a series of bases called codons. So we're going to unpack the term codon in a bit. We now move into the next process of protein synthesis, which is called translation. As I mentioned, the term translation refers to changing the language. So now we've got a sequence of nucleotides on mRNA, and I'm just illustrating that here for your reference. And that's, again, an mRNA molecule that's leaving and entering into the cytoplasm. At this point, it's important for us to recognize that mRNA contains what we refer to as, I'm just going to try and change the color of my pen, has these bases arranged in threes called a base triplet or a codon. So every 3 adjacent bases on mRNA are referred to as codons. So mRNA is made up of codons, and these lie next to each other. And these codons code for specific amino acids. So the amino acid codon or sequence is determined by the arrangement of these codons on the mRNA. Right. So the mRNA leaves, moves into the cytoplasm, and in the cytoplasm you have these structures called Rna molecules. And as you would see them, these are called ribosomes, which I've now further enlarged in this image. The ribosome is going to read the mRNA and read it in specific sequences called codons. And as it reads each codon, it will instruct a tRNA molecule, and here a tRNA molecule is represented by these green structures, to activate a specific amino acid based on the codon. What is unique to the mRNA is that an mRNA has a codon, whereas a tRNA, which I've illustrated here, has a specific anticodon. So that's the anticodon on a tRNA, and that often has a unique complementary nature to a specific codon on the mRNA. And that will determine which specific amino acid is brought into the ribosome. The ribosome continues to read each codon, instructs a tRNA to bring in a unique amino acid, and these amino acids are then held together by what we refer to as peptide bonds, and that chain continues to grow. And so we find that in this process that a polypeptide molecule develops, which is essentially a chain of amino acids, which will then grow and become significantly longer, and then compacted and reformatted into what we know as proteins. So as we wrap this section up, it is important that we recognize what we've just looked at. So we've had DNA that has produced a copy of mRNA. That mRNA leaves, it moves into the cytoplasm where you have the Rna molecules. The Rna molecules, or the ribosome, reads each codon. That's a codon. It will then instruct a tRNA molecule with the anticodons, which are here, to bring in a specific amino acid. So we refer to that as the amino acids. And these amino acids are activated by the tRNA. The tRNA will then transport or transfer that to a complementary codon on the mRNA. That's the new amino acid that has come in. The ribosome continues to read the next codon. It brings in the next tRNA molecule with a unique amino acid. These amino acids are then connected together by the peptide bond. And so we've just had a very quick overview of the process of transcription as well as so I'm going to rephrase this. That's translation, my bad, which is inside there, and transcription, which is inside the nucleus. And so as we wrap this section up, we've got to try and unpack the two terms as we end this. We've looked at protein synthesis as two complex processes. Transcription, as I mentioned, occurs in the nucleus, and it involves the production of mRNA. mRNA contains short sequences of 3 bases called codons, and these codons each code for a unique amino acid. When that process of transcription is completed, the mRNA leaves the nucleus and it moves into the cytoplasm. The DNA then regains its structure. The mRNA then moves to a ribosome, where the ribosome reads each codon and then instructs the tRNA, which are floating in the cytoplasm, to pick up amino acids. And these are done knowing that there is a complementary anticodon on the tRNA. The tRNA activates an amino acid, brings it into the ribosome, docks in with the codon, and we have the next codon being read, the next tRNA molecule bringing in the second, third, fourth amino acids. The amino acids connect with what we refer to as peptide bonds, and we find that once that mRNA has been translated, we have a chain of amino acids now formed, and we refer to that chain as a polypeptide. And essentially, we've looked at a complex process in 2 stages. Guys, you've done well. We've looked at a process now which is often difficult to understand. You guys need a break, freshen up. When we get back, we're going to look at how genetic coding takes place. Short break, and see you soon.